All right. Right. So good evening, everyone. Thank you so much for joining SynBioUK today for our second workshop of the series Fundamental Tools for Synthetic Biology. For those of you who may not know us, SynBioUK is a UK student-led society and community connecting academia, startups, and students passionate about synthetic biology. As the broader aim of this workshop series, we want to introduce you to a range of resources and tools that you can use to build fundamental skills in synthetic biology to the level that you can then apply to your own projects and research, hopefully. This workshop will specifically focus on understanding how protein structure prediction tools work and the context in which they can be applied and useful for your own research, as well as learning how to use more intuitive and accessible prediction tools. And Daniella will also walk you through some of these more accessible ones. And as I mentioned, this workshop will be led by Daniella Pretorius, a PhD student at Imperial College who works extensively with these tools and more generally in protein design and engineering. Before I hand it over to you, Daniella, I want to encourage everyone who's here in the meeting to follow along with Daniella's demonstration and to, of course, ask all the questions you have in the chat area, as well as at the end of the workshop, where we will also have some time allocated for you to ask Daniella all the questions you'd like. And these can be related to the specific tools or your own interest in how you can apply these tools to your own research. I also invite you to attend future workshops that we're organizing. In particular, the next one of this series is just before Christmas. So in a week, please join and engage with the SynBioUK community. And you can do this through our social media and through our Slack and through the feedback form that you will be given at the end of this workshop. Please give us some feedback about your experience. It's really just 1 minute. I try to make it as short as possible. And you will be directed to that as this meeting ends. And you will also get all the links to our socials and our Slack community. And so I'll now hand it over to Daniella to introduce herself and to kick off the workshop.
Hi, everyone. I'm quickly just going to share my screen. Cool. Is everyone able to see that?
Looks good.
Yeah. Okay, great. Thanks. So hi, everyone. Yeah, I'm Daniella. I'm currently a third and final year PhD student in James Murray's labs in the Department of Life Sciences at Imperial College London. And I do computational protein design, and also I am in the wet lab, and I'm expressing, purifying, and structurally characterizing the proteins that I design. So we'll start with the Make Your Protein workshop, right? Ooh, yeah. So we'll go over a little bit about how this workshop is going to be run, but just as a general point, I really want this workshop to be like a taster of protein design and engineering. I just want you to have a brief understanding of the field if you're not familiar with it at all, what tools are available and how you can use them for your research immediately. And I think the word immediate is like really important because a lot of these things take a long time to set up and it's really painful, but I want something that can be used directly within a day. You can have like some kind of protein design or engineering element in your workflow if that suits you. So yeah, we'll have a little intro and explanation on the field and techniques. Then we'll talk about what computational methods are out there and how you can use them. The meat of this workshop will be when we will all do a walkthrough with ARF diffusion, which is a machine learning model used for protein design. And we're going to make a protein binder and a protein motif that we'll scaffold a protein motif that I'll talk about later. And then finally, if you're interested in more protein design stuff and you want to go into more sophisticated advanced protein design, what do you do next? We'll kind of talk about that. So what even is protein engineering and design, just as a general concept? So these are two separate concepts, engineering and design, right? So protein engineering is when you modify or build on an existing protein, right? So one of the best examples of that, I think, and one of the most successful in the last few years is engineering about PETase proteins that break down plastic, like mutating residues in their active site to make them much better. And then on the other side, you have protein design, and this is where you're making new proteins from scratch, which this is mostly what I do. So a really good example of this is you have these mini protein inhibitors that were designed for the COVID spike protein. And these are actually in clinical trials now, but we'll talk about that later. But yeah, separate concepts, but I will say in the literature, people kind of use these a little bit interchangeably. And there is indeed a large gradient in the middle that you could kind of use either or both. But you know, it's kind of intuitive how you would do protein engineering, right? Like you're starting with the actual protein itself, right? But how do you build proteins from scratch? Like, how does that happen? So we'll talk about that a little bit. At its core, protein structure prediction, so what AlphaFold does, and protein design are two sides of the same coin. So protein structure prediction goes from a primary amino acid sequence to a 3D structure, right? But conventional protein design, you would use techniques like Rosetta, we'll talk about that a bit more to do this. You are taking a 3D structure of a protein, and you're finding the lowest energy amino acid sequence that satisfies this protein structure, or this protein backbone, as it's called. And this actually follows Anfin's hypothesis that for natural proteins, the native structure is the thermodynamically most stable structure that will occupy. So you want the lowest energy sequence. But, you know, this thing called AlphaFold was released, and it changed everything. So AlphaFold 2 was released in CASP. It's like a protein structure prediction competition that happens biannually, and it has completely changed the world. Just briefly, we won't really talk about the structure prediction stuff too much today, but just briefly. So AlphaFold, you're able to input your primary amino acid sequence, and using unique architecture, like machine learning architecture that hasn't been seen in the field up to AlphaFold 2, you're able to produce very high quality protein predictions, right? So it's been said that the single domain protein structure problem has been solved because AlphaFold will do it for you. But importantly for us, you get this 3D coordinates out of AlphaFold, which is your protein structure. But you also get these confidence metrics, PLDDT and PAE, that's what they're called. And it's really good because this actually allows the model to see how good it is, how confident it believes this prediction is. So PLDDT is a local confidence metric, and PAE is a global confidence metric. So we'll tell you if things like protein domains are going to be linked together or not. But yeah, so AlphaFold really took the world by storm. And in protein design and engineering, it's really split time into pre-AlphaFold and post-AlphaFold. So pre-AlphaFold, we were using this thing called Rosetta. It's a suite of different protein design techniques that you can use, but it's very difficult, and the success rates were very low because it was quite limited in what it was able to do. But most importantly, it required extreme fold expertise. You had to completely handcraft exactly what you wanted to say. It can't be scaled up. It's incredibly computationally intensive, and it's also a handcrafted algorithm. And by virtue of it being handcrafted, you're missing things that things like machine learning would implicitly be able to pick up on. And those are things that are really important for how proteins fold and what their structures are. So now after AlphaFold, it has led to a bunch of different deep learning methods, specifically for protein design being developed. And my favorite ones, and the ones that I really like, are using structure prediction algorithms that are either completely repurposed or retrained for the purpose of protein design. But what even does make a good protein design or engineering method? Like, what are we actually looking for? There are kind of three main things, right? So the first is fast convergence. So you're screening hundreds, if not thousands, of proteins at a time on your computer. So it needs to be fast. Ideally, every protein that you design, it should be on the order of seconds, not of the order of hours, because that's just going to take too long. It's not feasible. Another is you want to produce loads of diverse backbones, right? And when I say backbone, I mean the actual protein structure, right? Rather than the sequence. The sequence obviously indicates a structure, but just, unique structures, right? So diversity is really important in the game of protein design when you're approaching a specific objective. So the reason why is because it gives you more shots on goal, right? If you order 1 out of 10 designs, if you order 10 designs versus 2 designs, the chance of one of your 10 designs working compared to the 2 designs is much more likely. So you're playing a probability game and you want to increase your chances. And lastly, you want something that doesn't need a lot of context. So most of the time, and we'll get into this a bit more, you don't really know what you want in terms of sequence or structure. You have a general idea of some functional motif or something that you want to include in your protein, but you don't actually know exactly where. For example, you want to put the alpha helix on beta sheet. So you want the algorithm to really do the heavy lifting kind of in that department. Okay, it's great. So we know what makes a good method, but like, how do I know what actual method to use? A lot of them claim to be all of those things. So there's new papers with source code published every week on bioRxiv and arXiv. Actually, I think there's one quote unquote good paper being published every 2 days. But just keep in mind that experimental validation is king. You should always be ideally using techniques that have papers associated with them with wet lab work. You should be seeing crystal structures, electron microscopy structures, stuff like that to validate that this algorithm is actually capable of making what it thinks it's making. And another one is you want to look for good documentation and examples on the GitHubs for these things. So documentation making it easier to read the code as well, annotated things like that. And the examples will just be use cases that the people who make the code have given to you, and it makes things a lot easier to get started out. But okay, what is the best method? In my opinion, right now, protein diffusion models are the best in class method. And out of all of them, RF diffusion is by far the best method. But before we talk about RF diffusion a little bit more and we walk through it, okay, what even is diffusion, right? So a major example of a diffusion model that I'm sure a lot of you will be familiar with is DALL-E 2, right? And this is a text-to-image generation model. I'm sure you've seen the crazy images that you're able to make online and stuff like that. So the way that diffusion models work is you basically have your training data and you destroy that training data by adding noise. And then the model effectively learns to recover that data by reversing this noising process. And you can see it's like happening to our cat here. So in other words, the diffusion models can generate like coherent images from noise in this case. So you get cat from noise. And that has been translated to proteins with RF diffusion, right? So RF diffusion takes a structure prediction algorithm called Rosetta Fold. Rosetta Fold was the Baker's Lab, their answer to AlphaFold. They made a different architecture neural network that basically could do structure prediction, and it's very good. A little bit easier to do for things like this. But they basically take Rosetta Fold, which is a structure prediction algorithm, and they train it by getting their training data from the PDB of these proteins and adding noise to the protein structures. And then the model effectively learns to remove the noise, just like how we see in the image generation process. Nearly all of the protein design techniques that have come out actually take reference from image generation from machine learning that's been done before. And I mentioned about, experimental validation is important from the RF diffusion paper. Those mini protein binders that we were talking about that bind to the spike protein of the COVID variant, they actually were able to make these proteins and then make them into a trimer so that you end up with a more stable complex. And yeah, this is now in clinical trials. I recommend taking a look at this. It's really impressive. Okay, so that's great. We know what RF diffusion is. We know that that's currently, quote unquote, the best in class, right? But I want to use it now. How do I immediately use this protein design method in my research, right? So the problem with these things is that they take, they always, again, so most of these methods will require a GPU and a very good GPU at that. And they're also very difficult to actually install. Installing can be the most difficult process for these things. But we have these Google Colab notebooks. So what Google Colab is Google released these series of notebooks. If you're familiar with Python Jupyter notebook, it's quite similar actually in the way that it's structured. But when you log on, you get a free GPU for 12 hours. You basically instantly get a really great computer for 12 hours for free. It abstracts you from really complex code because it's a series of like almost plug and play type things. It's really easy to share, so you can share with your friends if you're having issues or your supervisors, etc. And it is like open source software is really prevalent in the protein design community. And this is one of the reasons why. So I would encourage to use platforms like this. So we're going to start doing the actual like meat now in the walkthrough. So I'm going to go on here. I'll actually post this link in the chat. Oh, sorry. Yeah, cool. So if everyone can go to this link, I just posted in the chat now, and it will take you, can everyone still see my screen actually? It will take you to this Google Colab notebook.
We can see your screen, Daniela. Thank you.
Cool. Okay, so yeah, I'll give everyone a second, but yeah, so here we're on the RF diffusion notebook. And there's actually, if you scroll to the bottom, it has an instructions page, and I'll just refresh this. Yeah, it has an instructions page, and it the documentation is quite good and clear, I think. But so the first thing we want to do is so here we are, we're in our Google Colab. You can actually see the code that's happening behind the hood here. So you can see that like it's installing like different models and stuff. So you can see everything behind the hood. If you do want to remove the code, you can just go back to form, hide code. But what we're going to do is you're going to press like the play button here. It's going to warn you that it's not authored by Google. We're just going to say run anyway. And this is the intensive part where we actually have to initialize and install RF diffusion. So that's going to take about 2 minutes. So while that happens, while that happens, I'll just show you through here. So as I said, it abstracts completely from the code. So what we can do here is we're going to specify exactly what we want for a protein. So you can name whatever you want the name of your run to be. You have your contigs. Contigs are a way that we can communicate to RF diffusion what we actually want to specify and what we actually want to do to the protein or make the protein become. That is what the majority of what we're going to go over in our examples. What I'll explain how contigs works. But you have that. You have an input PDB section if you want to put anything in. Iterations are kept to 50. That should usually be fine. This is just the number of times that you'll be going through the model. So if you have a very, for example, like a hard target, for example, like a beta barrel or something that's very hard to fold, you could increase the number of iterations. So you've got hotspot, which is specifically for binder design, where you want your binder to actually bind. A number of designs, like I said before, you want diversity, you want more shots on goal, so you want quite a lot of designs usually. And we'll go over these are kind of just like the output things, and we'll go over it in a sec. So I think we'll start here, right? So we have RF diffusion up, we have a little bit of understanding. Let's actually get to using it, right? So here's an example problem that I have here to make a protein binder. And I think that it's quite a common one, just to highlight what you would kind of be able to do, what you'd be thinking. So make me a protein binder between, 70 to 100 amino acids in length. And I want it to bind to the glucagon-like peptide 1 on a specific hotspot on this peptide that I specify, right? Just a quick side note, GLP-1, there is a drug that you may know that targets the receptor that binds to this, and it's Ozempic or Wegovy, the diabetic weight loss drug. So this is very important for things like glucose regulation, insulin, stuff like that. So after we make this, then everyone in the call can make a startup, I guess. But right, so we have this. How are we actually going to do it? How are we going to communicate with RF diffusion to do this? So like I said before, you have these things called contigs, right? And it is a way that you speak to the RF diffusion model. So contigs are a way to input very specific and varied requirements for protein design in a really concise and general way. And each contig basically describes a continuous polypeptide chain. So it's easier if I explain it here. Right. Yeah, so, oops, let's get this here. Yeah. So here I have the contigs chain. Oh, by the way, if anyone has like a small question or like something, or you can't hear me or you're not following along, just let me know. So we have our contigs argument here, right? What does this actually mean? So let's start with this. We have B10 to 35, right? Let's actually first, I'm actually going to get up the PDB structure that I want to use for this. So I'm going to fetch AIOL. Yeah, okay. So I'll just color by chain, right? So this is the peptide here that we see in blue, the GLP-1 peptide that we're actually going to be targeting, right? I can actually display sequence. Sorry, I think it's important for you to actually like see the structure before as well. Right, so here's our peptide, right? So I want to use this whole peptide for the kind of bits that are included in the crystal structure here, which is all of this, right? An important thing about RF diffusion is that if you're specifying a length of protein to use from an input PDB, you have to actually use the PDB indexing rather than like your own indexing of where it starts. So here, because crystal structures are kind of messy and just generally experimental structures aren't deposited very well, the first amino acid that we see is actually index 10. So that's what we have to use. So here, the first polypeptide chain that I'm giving to RF diffusion is the GLP-1 peptide. So I'm saying B because I want chain B. If it was if it was chain A, I would say A, right? So B, and then 10 all the way to 35, right? I want to use this whole thing. So I'm specifying the range of the natural protein that I'm trying to input, right? Then you have this colon. In RF diffusion, you have two different ways to like separate or distinguish contigs. I'll explain the other one after, but this colon basically says, okay, this is a separate chain now, right? Otherwise, it would link my actual peptide with the binder that I want to make for it together, which is no good for anyone. So this is saying, okay, this is a separate chain, this is a separate contig, right? And then I'm specifying here 70 to 100, right? So I said before, I didn't care what actual length it is. I just want it to be between 70 to 100, right? So what it will do is it will randomly sample this length, right? So if I have it going twice, maybe I'll get two different lengths. One will be 89 amino acids, and the other one will be like, 93 amino acids, right? So let's actually run this thing, right? So you can name it whatever you want. I'll just name this one GLP-1, right? Oh, an important thing, you can't have any spaces in the name because it will basically break RF diffusion, because it will try and look for like a file that's really difficult with these spaces. If you're familiar with Python, then you kind of know. But yeah, so let's see here. Yeah, so for our contigs, so we want chain B, which is our GLP-1 peptide, right? And we want 10 to 35. So I'm specifying the range, right? Then I want a new chain for my binder, and I want my binder to be between 70 to 100 amino acids, right? I actually have to put the PDB ID of what I want, and that's actually 3IOL. I'll keep it for 50 iterations, that's fine. And another really important thing is that you have this hotspot like requirement. So what does that actually mean? When you're making a binder, one of the most important, difficult things is for the algorithm to work out where that binder is actually going to bind, right? Or let's say that you have a huge protein and you truncate it and you end up with like these massive hydrophobic like kind of cross sections that are being exposed. The binder is probably going to bind there if you don't specify anything else, because it's this big hydrophobic like gap, right? The easier you make, like the more information you have, the easier it will be. You have this hotspot identification that basically will try and put the binder on these residues that you specify, right? So for us, the hotspot residues, and I'll show you here, are residues 28, so 28 and 29, right? And I'll actually color them here. So if we color them in red, and if I show the side chains, yeah. So this is our hotspot. This is what I want the binder to bind to, right? So that's 28 and 29. And the way that you'll specify that is really similar to how we did the contigs. It's from chain B, so we got to specify it's B. And we want to say 28 and then B 29. And you specify the amino acids like that. So for the number of designs, we'll just keep it to 1 just for the sake of the tutorial. And I think it's time to run it, right? So yeah, so you'll get this out, right? It'll tell you the job name that you specified. And for the contigs, you see it's B10 to 35, which is what we specified. That's cool. And then you see here it's 78 to 78. What that means is that it's randomly like chosen a number between 70 to 100, and it's 28. Aha. And here we go. So here's our peptide, and here is our binder. And you can see red is like a bad quality color, blue is a good one. And we can see that this is actually turning into quite a high quality like structure prediction, and it's improving over time. So with diffusion, you start from complete Gaussian noise, right? So you start with garbage, essentially. You just start with almost like atoms that are scattered randomly within a sphere space. And then you converge onto this really, really nice structure that fits our specifications. And also, this is incredibly fast. I mean, we've just done like 50 iterations in like 1 minute, 10 seconds. It's crazy. Okay, cool. Then you can have some fun with it. You can display the 3D structure and stuff like that. So if we can see it here. So here's our peptide, and here's the binder that we've made. These programs really like alpha helical structures, so they will usually get it, return it for you, like an alpha helical structure for you. You can change different things, so you can make it into a movie. I quite like these. We'll just wait for that to load. Yeah, so it's showing you kind of how, excuse me, how it started. So you can see it started from this kind of rubbish structure and then went all the way down. So currently we're coloring by chain. I would recommend coloring by PLDT. This is AlphaFold's output quality metric of local confidence. And it gives you a really good idea about how your design run is going. So if you look at it here, like I said before, red is bad, blue is good. You know, you start from this kind of rubbish structure. It almost, if it was predicted like that with AlphaFold, it would basically just be disordered, right? And then you end up having this really high quality structure at the end. Right. So that's amazing. But there's a catch, right? And the catch is that RF diffusion outputs only protein structure. It does not output protein structure and sequence. So these, so the proteins that we're seeing currently, the sequence, if we download them and open them on PyM or whatever, it's just populated with glycines. So we have to add in the sequence after. So protein MPNN is a very, very good algorithm, which is able to take a fixed protein backbone and get a very realistic sequence from that backbone, right? And it's really good at doing that. So here, it's great because everything's in the same notebook. I'm gonna make sequences for the structure that I've just generated, right? Okay, so I will just run that. Oh, sorry. That's fine. So yeah, so there are specifications for binder design here. I found that it doesn't matter too much, but we'll go over them. Yeah, we'll go over them a little bit. But basically we'll just put on initial guess. Initial guess means that it uses the like previous structures as a way to get better information now. Number of recycles means like how many times. So what's happening here is you're generating the sequence and you're taking those sequences, predicting them with AlphaFold, and seeing how similar those new structures are to your design structure. This is a very classic validation tool for protein design. So these recycles are the number of times that your structure is inputted back again to AlphaFold. So 3 is a standard. This is what, if you just use AlphaFold or CollabFold base, that's what you'll get. Yeah, so we'll run that again. It will take a little bit longer. I wonder who has the best design for today, because there's an element of stochasticity, of course, because it's random. So someone will have the best one. It's running AlphaFold and it should stop. And then we'll see the best result. Another thing with Google Colab, again, very similar to Jupyter Notebook, if anyone's used, anyone's used it, is that you can like run cells and it basically just like lines them up to be run. So when this cell has finished running, it will run this one afterwards. Okay. So I've done 8 sequences. Whenever you're running protein MPNN to get sequences, you always want to do a couple, just because there is there's usually people usually do 10 or so, and that's usually the best of what you get. Aha. So here we go. So we can see here's our peptide, right? And here's our binder, right? So oops, sorry. I'm going to show code. Yeah. So we can see what they've done is they've actually aligned the best structure prediction to the original model that we actually made. So what you're seeing here in gray is what we made up here, right? It's the model that, yeah, we basically wanted to make a sequence for it. And then obviously here, we're seeing the actual like structure prediction. We can see that's not very good, right? So that will require some fiddling. So we would have to go back. For example, are we targeting the right hotspots and stuff like that? But we'll carry on to the next example. But it's also, yeah, not terrible. I think that this probably needs like not just one design. It probably needs like 20 designs that something better will appear. But after you're done, I would recommend package and download your results. Like, it really is like you have your own computer, because you also have a disk space here, and that's where everything kind of gets saved. So when I called the PDB from earlier, 3IOL, it actually is saved here. So yeah, I would save that, and it will be saved in your results directory. But let's go on. Sorry, I'll move this here. Yeah, let's go on to the next problem. And the next problem is a motif scaffolding problem, right? So I'll just move that there. So what is motif scaffolding? Motif scaffolding is when you take a really valuable part of a protein, like an enzyme active site or something like that, and you scaffold it with other protein. And this other protein, you don't really care what it looks like or what it does. You just want it to be stable, and you want it to maintain the shape of that really important motif that you have, right? So here, so we've got this, sorry, yeah. So in this case, we have this scaffold site 5 from the RSVF protein, right? So this is our motif that we're going to use. And we want to take this site, and we want to scaffold it between the N and C terminal of the site with protein lengths of 10 to 40 amino acids. So it will be like a range of 10 to 40 amino acids. Again, I don't really care how long it is. The sequence of the sequence and structure of this site 5, and then 10 to 40 amino acids again. So kind of balancing it on both sides. So this RSVF protein, this site 5, it's a really specific like antigenic site on this protein. This basically the virus that this kind of comes from, or I believe it's a virus. I need to check. I'm not really sure on the physiology of it. Is that it makes like a lot of children, like young and older people, like very ill. But this is a highly immunogenic site, so it elicits like a very intense immune response. So the reason why you would want to scaffold something like this is to generate a vaccine, for example, just to give you like an example of why it may be important. So before I do that, let's actually get up the PDB, right? So in this case, this protein, we're just going to hide this. Yeah. Okay. So this protein has the PDB code 5TPN. Oh, we got to fetch it. Okay, so here it is. Okay, that's a little bit scary looking, right? So I know that site 5, right? Site 5 is in the region between, and I will write this down, 163 amino acids and also 181. Right, so I will color him in red here. So, and I will also show, it's really important to have a structural understanding of like what you're designing before you begin, I think. So I would encourage this always. So here is the actual site that we're scaffolding. We don't care about everything in cyan. We don't care about that. We just want to take this, right? So that is length 1.6. Oh, I've actually got it here. Yeah, length 163 to 181. Let's actually go over the contigs here again, right? So I said you're scaffolding it on the N and C terminal with protein that you don't really care. It's very similar to how we specified the binder protein here, 70 to 100, right? Here we're just doing 10 to 40, right? So 10 to 40 amino acids, whatever, randomly sampled on the N terminus. Then you have this forward slash. So forward slash for RF diffusion contigs means that this is part of the same polypeptide chain, but it's just another segment. Like I'm specifying another new thing for you to put here, right? So in the case here, the colon actually means this is a separate chain. These are not part of the same structure. This means this is the same chain, but I'm like, I want these to be combined together. So in this case, I want to take site 5, which is in the amino acid range 163 to 181, right? And I'm specifying A for chain A, right? Yeah, and then I have another forward slash and then 10 to 40, right? So let's see how this works when we run it through. It's crazy how you're able to do things so quickly. So we've set up RF diffusion. We don't need to do that again. That's fine, right? So in this case, let's start again. So I'll call this one site 5, right? So for the contigs, I want 10 to 40, A163 all the way to 181. And then I want 10 to 40 again. And for the PDB, that was 5 TPN. We'll go with 50 iterations. We don't need any hotspots. We're not making binders, so that's kind of useless for us. And again, just for the sake of things being quick, I'll just do one design, right? So let's run that. And we'll go down. There's always something super satisfying about waiting for the image. But yeah, you see here, so I've got 36 amino acids of diffused structure that it will generate for me. My site 5, that's going to be scaffolded. And then I have like 13 amino acids. So here it is. That looks a little bit small. We'll see how it performs. Yeah, I guess like these are also not optimal. They're some of like the examples that the RF diffusion team give for the local version. So I'm sure that you guys can think of course, more efficient ways to do this. But yeah, so we'll wait for that. This is also, I guess, considerably longer than the previous protein. Oh, no, actually, sorry, that was 59 seconds. No. All right, so I don't want to animate it. Let's display this and see what's happening. Right, so here we are. Okay, so if we color that by rainbow. Yeah, so rainbow just allows you to see from N to C terminus, it just colors it. If you have topologies that are kind of weird, it's good to see. So ideally, when you're finished with this, you'll generate the sequence for it and generate the predictions, and you will download your results, and you will actually do like an RMSD, so a root mean squared deviation comparison between what you've made and the actual active site to make sure that it is indeed in the same structure. But just by eye, just as we're going through it and as a checkpoint, that looks quite similar to me. That looks quite good. So I'm fine with that. We'll continue. Yeah, I'm actually going to do 2 sequences now, not more than that, but we'll keep the same number of recycles. Again, for the number of recycles, certain proteins are much more difficult to fold than others. So you can actually increase this to 12, for example. If you have a protein that has a really sophisticated topology, like a beta barrel, again, you'll need to up this. So if ever you're in a situation where the quality doesn't look good, I would increase the number of recycles and also the number of iterations of your RF diffusion model. Okay, here we are. Yep, so protein MPN and AlphaFold. Protein MPNN runs very, very, very quickly. So the determining factor here is AlphaFold itself. Okay, so this was our previous best result for the GLP-1 thing. Let's see here. All right, that is actually pretty good, not gonna lie. So that's better than my last one. I wonder how it is for everyone else. And again, I would download that. So let's download this one to see. right, so yep, I'll just save that. All right, so if I go to my downloads folder, so it'll have the name of my, yeah, here we go. So here's my protein, right? If you want to view the PLDDT, which is the local confidence metric, you can do that by going to, which should work, coloring by B factor. Yeah, so for B factor, it's actually the inverse of what we usually think. Usually, like blue is good, red is bad, right? But in this case, it flips it in PyMol for whatever reason. So yeah, red is good and blue is bad. So here we actually have a very like good structure. And what I would recommend, yep, that's good. Yeah, in this information here as well, you will have the output confidence metrics and their graphs. Yeah, I need to look at it properly, but yeah, the output and the confidence metrics and graphs, and you'll be able to look at that properly. But yeah, I hope that everyone was able to follow along with that and enjoyed that. So that was a really kind of simple way. I just wanted to show you guys, if you weren't sure, these things are out there and they're incredibly high quality. So I would really recommend using them. But if you are more interested in advanced protein design and that's where you're going, and if this was like a too low level for you, then I would recommend do a GitHub local installation. There's plenty of information on the respective GitHub 41 on there. The only thing is you need a GPU, and it has to be a pretty good one, like I said. I recommend using your university high performance computing cluster if you have access to that. If not, you can actually use the like Amazon Web Servers or Google Cloud Computing. You can do things on that are quite sophisticated. And just as a kind of finishing note for everyone, I said RF diffusion was the best, but here are a couple of other things, and here are their respective GitHubs as well. Yeah, so RF diffusion for realistic backbone generation, protein MPNN to generate sequences for backbones. These together are the best. You also have this thing called protein generator. It actually diffuses protein structure and sequence, but it's not as good as these two together. Especially on longer proteins, it really isn't as good, but that's out there and it's quite fun to play with. You have collab design, where people are repurposing structure prediction algorithms like AlphaFold and using them for design like that. And then I don't know if anyone's seen, but Chroma has been released. It is like Meta's resultant kind of like generative protein design algorithm. It is okay, it's very fast because it doesn't use a structure prediction algorithm, but it is not as good as these guys for sure. But it is very cool, and it has a very snazzy, well-documented code base. So that's what I have for the tutorial. I will quickly, shall I stop sharing?
Yeah, that sounds good. Wow, thank you so much, Daniella. This is really cool stuff. I appreciate that, you know, many people were following along on their own devices and may not have had enough time to write their questions in the chat. So this is your
sorry,
time.
I thought someone was asking for the contigs and I didn't specify again. Sorry, I couldn't see the chat.
Yeah, no, yeah, I think thank you for people for providing that. Yes, you will be able to access the slides and recording later. We'll aim to get this up within a week.
Yeah, like where will this, because the slides will also be available, right?
Oh, yeah.
Yeah, okay, cool. Yeah.
So anyone just feel free to ask any questions, and these can be related to the tools Daniella showed today or anything that you're interested for your own research, really. So it can really be anything you're interested in applying protein prediction tools.
So I see someone asked, how did you learn to use all these programs or software in the beginning? Did you learn on your own, or was there someone to guide you? So my background, sorry, maybe I should have started with that. My background is I'm a biochemist. I did biochemistry at Imperial, and I had zero computational experience coming into this PhD, like nothing. Like I hadn't even have done a Python course. And my lab is not a specialist in this field. We are like a structure biology lab, which is where my validation of the structures come in. But basically like I did it on my own, and I will say it's easier now than more than ever to do these kinds of things on your own, mostly because there's so many open source things like these collabs where you're actually able to play with things and get an understanding. There's also a series of really good like webinars, machine learning for protein design by like the Boston Protein Club. I can give you I can give you like all these things that I'm saying, Tomas, and maybe they can be published later. Okay, great. Yeah, phenomenal. And I think also a lot of the papers are really clear about how they design things as well. So I think the advice is no one was there to guide me, so maybe it took longer than it probably should. But if you are interested in this stuff, just start, and there will be so many people willing to help you. Like I'm going to MIT next year to one of the main guys for the RF diffusion network, and I just messaged him on Twitter, and like now I'm going to like collaborate with him for a few months there. So if you are confused, reach out. Reach out to everyone. You can reach out to me too. Can you go over scaffolding again and its applications? Yeah, so scaffolding, it's like a really classic example for protein design. The point of a scaffold is it's like really stable, right? So sometimes what happens is you have like a base protein, and it's really stable, and it's like high thermostability, very resistant to mutations, stuff like that. And you can add functional groups to it, whether that be a spike tag catcher, whether it be a loop, whatever you may be doing, right? In this case, like you can take a functional site, like you can take the heme binding, residues from like myoglobin, for example, or hemoglobin, and you can generate a protein backbone that will be able to keep those residues in the same place and keep them with the same rotamers, right? Because an important thing about like proteins and amino acids is that, of course, you have the amino acid, but then it also has several rotamers that it can actually take. I hope that made more sense for the scaffolding. You're just generating whatever kind of protein to stabilize your functional motif. And motif, I guess I also use that, but it's just any like functional part of a protein. If you could go back to Imperial Biochem undergrad, would you start learning computational skills earlier? Yes, like 100%. I would absolutely do that. It was a complete mistake. I think it's a big disservice. When I was at Imperial, I started in 2017, and there was one Python module. There was a bioinformatics course for third year, but it was like a selection process who got that. And I think that start as soon as possible. Not gonna lie, I think that like with ChatGPT, coding is easier than ever now. The important thing is having an intuition and knowing how to set these things up, and working with, for example, the computing cluster and stuff. That's probably much more valuable for what I can bring than my actual coding. And honestly, I outsource a lot of the boring stuff to ChatGPT now. I even have a paid subscription, right? So I will just get that to do the boring grunt work for me. But I had to go through the painful process of learning it first. But yeah, start as soon as possible. I think it's like Data Camp has like a really good course on that, like a generic Python course. I will also say that there is a professor from Harvard who has a stellar machine learning course. Andrew Ning, I think. I'll give the link to Tomas, and we can share that with everyone. But I would recommend do a machine learning course and do a Python course, because not enough these days as well to just do a Python course, as unfortunately. Do you know any instruments to predict enzyme activity and their properties, such as thermostability and pH stability? Maybe with using deep learning. Maybe So when you say instruments, do you mean like in the lab what you would use to do that? Or is it like a experimental instrument that you would use? Oh, like a bioinformatics tool. So what I would say is predicting thermostability, right, and pH stability is a little bit of a dark art. There are a lot of bioinformatics tool online that will do that for you. What I would recommend is protein MPNN, which is the algorithm that I spoke about previously, which takes a backbone and generates a sequence for it. That has been shown to generate ultra stable, ultra thermostable, and pH stable proteins. In fact, with protein MPNN and RF diffusion, you were able to specify, it is like an argument within the program if you download it locally, you're able to specify the pH range that you want this protein to be in. You're able to specify the isoelectric point, which is incredible, right? So therefore the pH stability. Also, they have a whole paper on how they make things more thermostable. So they did that on a TEV cleavage protein, so a TEV protease, and they also did that on myoglobin. They kept the same the important residues the same and redesigned the backbone, and they got like really great results. I hope that helps. Yeah. I also I can maybe even find the paper now. Yeah, here we are. And all of these papers are like on bioRxiv. Yeah, here it is. I'll put this in the chat. Does anyone else have anything?
Yeah, you know, I'd like to ask one myself if that's possible.
Oh yeah, sure. Shall I there's one more here and then we'll get to your one.
Yeah.
What do you think is the most important to learn first and advance in microfluidics, nanofabrication, coding, lab techniques? Whoa. Which do I think is the most important to learn first and advance? And it depends what you want to do, I guess. I'm not familiar with anything to do with microfluidics or nanofabrication, but I think it's not about like learning first. It's like these things actually, I can only speak to coding and lab techniques, but these things fundamentally are like the tools that you use as like a scientist, right? And I would say that like me going into the dry lab and learning how to code and doing these things makes me a better scientist, and therefore makes me better in the wet lab because it makes me think in a more logical, rigorous, like kind of way, right? So I think they're just all skills. It depends what, I mean, if you're working with like, I don't know, Yuval Alani or whatever, or doing stuff with microfluidics and stuff like that, then of course it's probably microfluidics. But I guess it just depends. But I think everything, it's like a sum of all parts, like everything adds together. These programs can show different predictions with the same input. When you say these programs, what exactly do you mean? Oh, so when you say this example, sorry.
I mean, what we were doing right now.
Yeah. Oh, sorry. So when you say like what, like when you ran it, like it gave different outputs.
Yeah, that's my question.
Yeah. So so that's kind of like one of the best bits about it. You will get a different output every single time. And it adds to that diversity that I was talking about before, right? The reason why for this technique is because you're starting from complete random noise, right? And that's randomly initialized. So what you end up with is like where you start influences where you ultimately end up. Of course, you'll end up with things that are kind of similar more often than not, but they will be different. Yeah. And that's why if you run 10 or 20, all of those 10 and 20 will have different structures.
Yeah, if anyone has any more questions, feel free. If not, I want to ask my own question, really. Regarding the quality scores that you mentioned, for example, PLDDT, the thresholds that are considered low, medium, and high quality prediction, are these the same across all protein or protein regions, or can they vary according to the structure and properties of the predicted region? So maybe the number of interactions within the protein or the specific amino acids in that region, or is it just the same kind of threshold across?
So you're saying like PLDT, oh, sorry, I just activated my Siri. PLDT ranges from 0 to 100, right? 0 being like a complete garbage, hot mess. 100 being like, this is absolutely crystallographic quality, like structure for this protein, right? Like 100% confidence. If the thresholds for what is good and what isn't, I would say is the same for all of them. And that is above 90 is experimental quality prediction output, right? I would expect that that means that because AlphaFold is also trained on nearly 100% crystallographic crystal structures, right? That means that the probability of me expressing this protein, purifying it, and then making a protein crystal out of it, and then finding the structure, it's like really, really high confidence that that crystal structure is going to correlate with the structure, right? Because we haven't solved like what proteins look like in their regular lives. But basically, yeah, so above 90 is like experimental grade quality. Above 70 is good. Below 50 is awful. So when I'm designing, I always have an output threshold that like it needs to be above 70. Just as a general kind of thing, yeah. Does that answer that a little bit more?
Absolutely, yeah. Thank you. I think we have another question.
How do we ensure the specificity of binding between the target and the generated peptide? Right. So when you say specificity, do you mean that like you're not going to you mean you're not going to generate a binder that is like promiscuous, for example, and do other kinds of things and bind to other proteins? Okay. Yeah, that's a little bit more difficult. I think the first thing that you can ensure that like if this protein like is very well predicted and binds like with like in this case for binding you would use PAE, right, which is the global confidence score. We don't have time to go into it today, but there's a lot of description about what it means and how you would analyze it online. But you would want that to be as high as possible, right? Because if that has a super high affinity, then that's great. And that's pretty much only going to bind to your partner when it's there. If you're worried about things cross-talking, if you're worried about specific candidates, what I would do is you can do like a negative design strategy. So you can predict or design with with other candidates there, right? So you have the peptide that you want to bind to, and then also like 7 other peptides, and you're optimizing so that it binds really well to your peptide with a really high affinity, and it binds really badly to the other peptides. But I think the best way of doing it is make sure that you design something that binds with really high affinity. Is it possible to actually predict binding positions for RNA binding proteins on RNA, knowing that most RNA motifs are generated? So that's really interesting. I do have a DNA binding protein project. And basically, I'm not really familiar with RNA, to be honest with you. I'm not familiar with nucleic acids basically at all. But currently, can you predict that? I don't know what people have done in the bioinformatics space that works specifically on RNA and DNA and binding sites. I don't know about that, but there was a paper that was published. It was called Rosetta Fold 2NA, where they took the Rosetta Fold algorithm, but instead of just using proteins as a training data, they used protein DNA complexes that were deposited in the PDB, right? And they trained it, and it was actually pretty good. But now AlphaFold and Rosetta Fold have released versions which are all atoms, right? So currently you can pretty much only do amino acids. The new all atom versions can do ligands, DNA, RNA, small molecules, blah, 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 everything. So can you accurately predict binding positions for an RNA binding protein for RNA? I would assume that when the code is published, which it hasn't been yet, for Rosetta Fold all atom, you would be able to, well, I would screen variations of RNA with your protein, and you would be able to see in that way. But I'm not sure how other people in the field have kind of tackled this on a more sequence-based, because I think important to say I'm a structure-based person for protein design, and there's a lot of people working with like sequence-related stuff, but it's not really what I do.
All right. You know, I might give a few more moments for anyone to have some very last minute questions, and then we will wrap it up in just a bit.
Cool.
And then just a reminder to everyone, and thank you, Daniella, for offering to provide more resources on this after the workshop that we will then distribute to all our participants and anyone in the SynBioUK community to then inform themselves more on protein prediction tools.
Cool. Yeah. Hopefully. I didn't quite understand what's PDB. Oh, sorry. So when I say PDB, so you have well, I'll quickly share my screen. Right. So when people experimentally characterize proteins, they go into the PDB, and the PDB stands for the protein data bank, right? And basically what it is like you have your specific code for your protein. This is the protein that I work on. And it has like the actual like structure information of the residues. So, and the file output is called a PDB. So this is an output PDB like structure. And you can see here it has like structure, like if you look at it, it's got all the like positional information for all of the atoms, but it's called a PDB file. So it's just the experimental structure of your protein. I guess it's not just experimental anymore. It's like any protein structure is just called it's got the extension is .pdb.
All right. So I think we'll wrap it up here if nobody else has any burning questions. So yeah, thank you so much to everybody for participating in today's workshop. I hope it's been a useful introduction to protein prediction tools, and that your understanding of them can then be applied to future work or research that you undertake in synthetic biology or anything else you're interested in. Please let us know your experience in this workshop through the feedback form that you'll be directed to after the meeting. I made sure that it's very short, so it shouldn't take you more than just 60 seconds to complete it, and will significantly help us deliver relevant and engaging content to you in the future. And of course, I also want to extend a massive thank you to you, Daniella, for sharing your knowledge and experience on this topic, and for making this workshop so engaging for all the other students. We have one more exciting workshop lined up, and it's going to be next week on benchling basics. But SynBioUK is also planning other really exciting initiatives next year. So please sign up to our mailing list to stay tuned. And you can also find us on Instagram, LinkedIn, our Slack community. And of course, you can reach out to us via email as well. And again, all these links are in the feedback form. So make sure to check out the feedback form and to check these links out. Thank you, everybody.